Thank you very much, Chairman, for your very good introduction. And sorry, but that was my previous short biography. I actually now work for the University of Melbourne. I moved 3 months ago. I'm sorry that it was the old one. So I'd like to thank Fabio Montossi and the organising committee for inviting me to speak. I really appreciate the opportunity. And I have 3 wonderful co-authors, and we decided to go alphabetical with the authors. Joanne Hughes is a young PhD student, and she's very good, and we were very pleased to offer her the opportunity to go as the first author. So I'll be talking about a structural approach to understanding the interactions between colour, water holding capacity and tenderness. So I'll be giving you an introduction, of course, and then I will be talking about structural determinants of water holding capacity and water loss, and comparing the differences between raw and cooked mussel. I will talk about the structural interrelationships between water holding capacity and tenderness, the influence of structure on mussel colour and light scattering, and thank you to Suman for giving us an excellent review on the biochemical aspects of colour, whereas I'll be talking about the structural influences on colour. And also I'll give some conclusions and further suggestions for R&D. So introduction, firstly, quality. We've talked about it lots and lots and lots over the years. Visual. What I'll be talking about today is the colour aspects in terms of what we see in the meat on the shelves, the softness or texture, and also the water holding capacity. For sensory, I'll be talking, in terms of the cooked product, I'll be talking about their tenderness and the juiciness. There is obviously many other meat quality traits such as shelf life, flavour, etc., but I'm focusing on these quality traits in terms of the structural determinants. In terms of these visual and sensory traits, these are all affected, as we know from many years of research, by the rate of pH and temperature fall. But also, all of these traits are affected by ultimate pH. They're all influenced by the structure of mussel, partly through the effects of pH, rate of fall, and ultimate pH. And there is certainly interrelationships between these traits. So in the top picture there, you actually see some beef. Now the top longissimus mussel in that picture is actually what we call high rigor temperature or heat toughened or heat shortened beef. So it's actually PSC-like meat in a beef carcass. The middle one is normal and the bottom one is what we call dark cutting or high pH. And the bottom picture, of course, is of some pork muscle showing DFD or dark firm dry, high pH meat, normal, and some PSC meat. And as we know, these conditions showing the range from pale to dark are what we're talking about in terms of the influence of rate of pH fall and also ultimate pH. So also in terms of these quality traits, it's important to remember why they are important. So obviously weight loss, if we have loss of water through purge in the vacuum bag or through drip loss, that is a problem. And also the economic loss of dark cutting. In Australia, if we have a dark cutting beef carcass, the processor loses $400 per carcass. So it's a big cost in our industry and runs at about 10% nationally. That we also, we must ensure quality for a sustainable meat industry. If we don't produce quality, people will stop eating meat. Also, we do get premiums for high quality, and this has been shown in research papers on willingness to pay. So Meat Standards Australia graded meat gets a higher dollar for the producer, for the processor, as well as for the retailer, making sure the consumer gets a better quality product. So this is also why these quality traits are important. So what could be new? We actually have had many presentations over the years on water holding capacity and tenderness and colour, many fantastic reviews. So in our review, we did focus on not just presenting what's already been presented, but some new material which you will see in my presentation. So the variations in water holding capacity, tenderness and colour have previously been explained from a structural basis by Offer, Tornberg, and for colour I forgot, of course, also Howard Swatland in Canada. Biochemical and metabolic understanding, Huff-Lonergan, Scheffler, and also Mancini and Hunt in terms of colour again. And it's also been modelled by Eric Dransfield for tenderness, Conjoin, who's in the room, has done some great work in this area, Offer, Tornberg and Van der Schmaan, who presented last year. So modelling has come a long way, but often we use modelling to identify gaps in our knowledge, and especially we've seen some gaps, I think, in the cooked meat area. So there has been much research over the years on raw meat, but in fact we actually eat cooked meat. So the focus, we think, should be more on what's remaining in the muscle and what's actually happening after cooking. So it's still what could be new, making sure that we are producing something new in this review. In fact, there's often a poor correlation between water holding capacity in raw meat and the water loss during cooking and also with the juiciness. Why would this be? And I will be answering this to some extent. Also, in terms of genetic influences, sometimes there is genetic and metabolomic studies where they find complex enzymes and metabolic systems which are proposed genes which have an influence on meat quality. But sometimes we wonder if there's actually a fundamental mechanism behind some of these genes, such as pH and, of course, the metabolism associated with that, as in the AMP kinase work that David Gerard and his group have been involved in. So in a myofibril, what is there in the myofibril? What is there besides protein in the myofibrillar structure? It took me ages to put these little dots of light blue that say H2O, but there's a lot of water in the myofibril. And in fact, in a muscle cell, the volume of the muscle cell is 80 to 87% filled with myofibrils. And yet, the chemical composition of a muscle cell, a muscle, is 75% water. So there's an awful lot of water there which is actually within those myofibrils. So what I will be talking about is any changes in this myofibrillar volume and lattice are very important in terms of how much water can be held within the structure, which influences the water holding capacity and changes during cooking and tenderness. So water is a part of the structure. We think it's the third element of structure and tenderness. It allows diffusion and interaction of substrate and enzyme. It also determines plasticity, rigidity and gelatinisation of the insoluble proteins. Now, water holding capacity is important for visual and sensory acceptability and also for economic reasons, but it's also important because of its role in moulding muscle structure and the consequent effects on quality. So this shows some different pictures of water holding capacity. This is the one that I showed before, going from DFD, which basically doesn't lose any water post-slaughter, post-rigor, to PSC, which can lose up to 15 to 20% of its weight in pork. Again, DFD, normal PSC pork with a lot of water coming out in the PSC pork. This picture here shows some beef which has actually been through high rigor temperature or heat toughened or heat shortened, and a lot of water is actually in that vacuum bag, as you can see in the bottom there. This is a great picture that I love. I think it actually came from the Netherlands. Bob Kaufman gave it to me, showing a study where they did a lot of different bags measuring drip loss in all of those, lots and lots of samples of some work they were doing in the Netherlands. So another way to measure water holding capacity. So basically water holding capacity is the water loss from raw meat, not from cooked meat. So there's really, we think, 4 structural determinants of water holding capacity in raw muscle, and these are myofilament lattice spacing, membrane integrity, cytoskeletal linkages, and extracellular space development. And I will go through each of these now. So firstly, in terms of the driving mechanism of drip, whether we have transverse, which is across the diameter of the myofibril, or longitudinal shrinkage of the myofilament lattice, we actually drive water out of that structure. So here we see the actin filaments, thin filaments, and the thick filament in the middle, and as that shrinks either lengthwise or in diameter, you're actually driving water out and fluid is lost from within that myofibril where most of the water is held. So when the myofilament lattice spacing is reduced, water is expelled from within the myofilament lattice. So therefore the final lattice spacing, and hence the drip, is controlled by anything that shortens or lengthens the sarcomere or changes the diameter. And these factors include sarcomere length, pH, myosin denaturation, and the Rieger bond. So in terms of reduced myofilament spacing, we do get reduced myofilament spacing due to shorter sarcomeres. So this shows some results from Peter Perslow and Agnes Pruss, which they haven't published yet. And here we see, this is from porcine psoas, and the vertical axis is percent weight loss, and the mean sarcomere length to the right-hand side is down up to 2.6 to 2.8, so quite long, and not as much drip loss as down this end, where you have more drip loss at the shorter sarcomeres. So the shorter sarcomeres, more drip loss. But furthermore, it also shows that you get this much drip loss in the line at 2 days post-slaughter. At 3 days, it's like this, and by 6 days post-mortem, the drip loss, certainly when it's shorter than 1.8 microns, is starting to increase by 6 days post-mortem, even more. So the myofibrillar shrinkage only really results in muscle fibre shrinkage, if there is connection to the cell membrane. So what we have is our myofibrils, which are in the muscle cell, and they're all linked at their Z line, all linked to each myofibril, linked out to the costomeres, out to the sarcolemma, through these proteins such as desmin, ankyrin, and other proteins that link right out through to the sarcolemma, the outside cell membrane. This is the cytoskeleton of the skeletal muscle. So we see more of these proteins shown here. There's some certainly desmins in here and also in here, which is the ones we're talking about now is we're about to talk about the proteolysis of the proteins here and between the myofibrils. So these cytoskeletal proteins actually form strong intracellular linkages between the myofibrils and also out to the plasmalemma. Sorry, yeah, plasmalemma, not sarcolemma. Sarcolemma, yeah, same word. So this is a hypothesis that Christensen and Perslow proposed in 2001, where they basically said that here's your myofibrils within a muscle cell initially, and then if these myofibrils shrink, if they're still attached to the cell membrane, the whole cell shrinks. But when you have proteolysis, so breakage in that desmin, etc., those other proteins, these can shrink, but the muscle cell can either not shrink or can actually swell because you've had proteolysis of those proteins. This picture here shows the water flow rate from intracellular to extracellular compartments. This is hypothesised, not actual data. So you see this increase in flow of water initially out to the extracellular compartment. But as proteolysis proceeds, which is this dotted line here, you see this movement of water back to intracellular, and the muscle fibre can even not only go back to it where it was before, but can even swell during ageing because of losing these linkages. There's also the formation of drip channels and the change in membrane integrity. So firstly, in this one we're showing, this is in porcine psoas major muscle again, time post-mortem 0, 6 or 9 hours post-mortem. And this is the measure of impedance, which actually measures the membrane integrity. And in a low drip loss muscle, there's very little change in the impedance. It stays at 70 from 0 to 9 hours post-slaughter. In the high drip loss muscle, you'll see that the impedance drops from 70 down to 10, showing this loss in membrane integrity, which means that the water can flow out of the muscle because of the membrane starting to disrupt. The other thing this picture shows is here, cross-sections of the porcine muscle cells, and you'll see in the low drip loss muscle, not a lot of change between 0 and 9 hours, a few gaps starting to appear but not too many between the muscle cells. In the high drip loss muscle, you'll see there's already some gaps between cells at 0 hours post-slaughter and by 9 hours post, are quite big gaps. And really what happens is this water flows out from the cell and then comes down through what are called drip channels, and can come down these drip channels and flow out actually as drip or water from the muscle. So that's the 4 mechanisms I talked about. So now we move on to water loss during cooking. So the structures and mechanisms which are influential in water loss during cooking are not as well studied as those that have been studied in raw mussel. And they are certainly to some extent or large extent different to those in raw meat. The correlations between water holding capacity and cook loss or juiciness are shown here. So Margaret Esling and her co-workers have shown that at 80 degrees Celsius cooking, only 20% of the variation in cooking loss is explained by the water holding capacity of the raw mussel. If that temperature drops down to 60 to 70 degrees in cooking, there's certainly more of the variation explained. Also, Winger has a very good chapter in a book on juiciness. And he talks about the lack of relationship between sensory juiciness and objective measures of water holding capacity. So we need to understand this, we need to understand the differences between raw and cooked meat shrinkage. So firstly, we can look at the relationship between sensory juiciness and cook loss with temperature, which is reasonably well correlated. So as the temperature goes up from about 45 up to 90 in cooking, we actually have this increase in cooking loss from about 5% right through to close to 35%. Consequently, as a consequence of that, the juiciness of the meat, actually this is from Martin's, drops from about a high score, very juicy, down to a low score for juiciness. So certainly during cooking, the meat can lose a large quantity of its mass in a time and temperature dependent manner. And certainly cook loss and sensory juiciness are correlated. So now we'll talk about some of the structures and proteins that are involved in the shrinkage of meat during cooking. The time's disappeared. Doesn't matter. The time's disappeared, that's okay. So firstly, the role of collagen. The following I found a few, I had several in and just left one in the end. The following statement can be found in many textbooks. I'll need you to help me remind me of the time. Yeah, thank you. Can be found in many textbooks and papers. Toughness and cook loss due to tension in the perimysium and endomysium from thermal denaturation and shrinkage of collagen. So the shrinkage in collagen causes the toughness and cook loss. But certainly the studies that have shown no correlation between collagen content and cook loss, such as the really classical study by Rhee et al. In 2004, which includes Tommy Wheeler as a co-author. So there must be some other proteins involved in this shrinkage during cooking, which I will now talk about or propose. So some of the changes in cooking loss with temperature, this is a graph from Eva Thornburg from a paper in 2005, and it shows on the vertical axis cooking loss. The top line is whole meat, the next dotted line is hamburger, and the bottom line is sausage, and across the x-axis is the temperature. So it goes from about 45 through to 80. So what you see, the only difference between these two lines is at 65 degrees. For all the others, the standard error of the difference is actually the same. So what you see is this increase in cooking loss up to 60 degrees, and then this much more substantial increase in cooking loss between 60 and 75 and 80. But basically, they're following along the same lines, and certainly saying that in hamburger, you can't have a roll from connective tissue because it's been destroyed. And in fact, Hannah Bartram from Denmark has also used NMR to show that in hamburger muscle, the water water mobility is the same as in raw meat. So again, suggesting the lack of not as big a role of collagen as we thought. So this, at 65 degrees, this difference is probably cooperative shrinkage between collagen and muscle fibres in the hamburger. And this is the temperature, this cannot be only collagen shrinking that's causing this water loss. So what is shrinking apart from collagen? Oop, I thought I had another one there. So we actually undertook an experiment. We looked at the changes in a muscle cell, an individual muscle, fragment of a muscle cell, sorry, during heating from 25 to 95. And this one here just shows some muscle, little muscle strips cooked at 65 degrees through to 90 degrees, 65, 70, 80 and 90, and straight away you see the shortening in this little strip, and also there's a decrease in the diameter. So this was some work we did, that Janet Stark did with CSIRO before we did this study. So we had 6 semi-tendinosus beef muscles. These were homogenised into muscle fragments, and then we placed them on a heating stage on a glass slide, and we did this a number of times. And we've actually taken our movies. So we observed this under the confocal microscope, the actual individual muscle cells as they were heated from 25 to 95 degrees. We've done this for a number of different muscle fibre fragments. So I'll just show you the movie now. Hopefully it'll work. Where's the man gone? Oh yeah, here we go. Accept. So this is showing as it's heating up to and around 50 to 55, which is coming to now. You see the shrinkage in diameter of the muscle cell. And then there's a quite discrete and separate event around 80 to 85. You see a shrinkage in the longitudinal length of the muscle cell. So it's going to show it again. So there's certainly some basement membrane there and possibly some endomysium, but predominantly what you can see, I think, is a shrinkage in these myofibrils and the proteins in the myofibrils. And what's really interesting is how discrete and separate those two events are. So you see it happening transverse at a very different temperature, around 50 to 55, compared to the longitudinal shrinkage, which happens around 80 to 85. Of course, the other thing that happens when it shrinks longitudinally is it actually gets wider in diameter because of this isovolumetric nature of a muscle cell. You seen enough now? It'll just keep looping on forever otherwise. So we've also taken a number of these images, and so not just the one muscle fibre, which you see in that picture there, but taken a number of these and actually analysed them for their changes in volume. And this picture here, which is in the paper that's in the Meat Science edition, actually shows the volume change in those muscles, which we calculated as you go from 25 right through to 90 degrees on the 90 degrees on the heating stage. And this is the pictures at 25, 55 and 75. So obviously you're actually following the one muscle fibre, not lots of fibres, just one muscle fibre through as it's heated from 25 to 90. And what you see is this volumetric change, not too much change here. When you get to 40, you actually start to get some volume change. And the steepest part is from down to here, down to around 65 or so. And then it flattens out down around 65 to 70 and right through to 90 in terms of the change in volume. Nope, this one. So certainly the major change in the state of water, this was demonstrated by Mick Lander using, I think it's either NMR or FTIR, is around 65 degrees. And this is actually where generally people say collagen denatures. And so you don't really see, in terms of the volume change in this muscle cell, much volume change at all. So I think that's very interesting also. So what time? 15, thank you. You're a good signaller. So the proteins that we hypothesised to be involved in this lateral or transverse shrinkage, which is the shrinkage in the diameter, there's a few that we've talked about in our little group. The first one is possibility of collagen. Now, collagen denatures around 65 to 67. Some of you might have a different number for me, which I'm happy to talk about. Myosin generally denatures around 38 to 54 degrees, and I quoted this from Ellen Conjoin's paper. That is, in fact, pH dependent in terms of what temperature myosin denatures at, and I've pointed to the bits of myosin in here. Desmin will also denature at around 46 degrees, and this is from Caroline Barron's work, which is unpublished results, and I think Caroline's down the front somewhere. There she is. Yep. So the other possibility is Desmond. But we think in terms of this possibility of cooperative shrinkage between different proteins, because in the volume change, you actually see this start at about 40 and it continues to change in volume. But we think the main one that's happening at around 55 is probably myosin. And because certainly when myosin denatures, as Gerald offers are very well shown, when myosin denatures, it pulls the whole of the myofilament in closer to it. So it's quite logical that myosin might be the one that's actually causing that shrinkage in diameter at around 55 to 60. The proteins are hypothesised to be involved in the longitudinal, we don't think it's collagen there. The proteins hypothesised to be involved in the longitudinal shrinkage. Now this was happening, probably started around 75 and was finished around 85 degrees. So again, collagen denatures around 65 to 67, so we don't think that has a role here. Actin denatures around 70 to 83, again, pH dependent from Burdasong and Song in 1990. So we think this is certainly a strong contender for that shrinkage at 80 to 85. We also thought it might be titin. I kept saying, must be titin. I've been well trained by Marion Grieser at University of Wisconsin-Madison in the USA to love that molecule titin, big protein in the muscle cell. But in fact, we don't see a change in the, we actually did fresh and aged muscle fragments, and we didn't see a change with ageing. Now we know titin degrades through proteolysis and goes down to the doublet and the triplet. So because we know titin degrades with ageing, we don't think that titin is the target molecule, but it certainly may be involved. So again here, we think it's probably actin, but it might certainly be cooperation with other molecules. 10 minutes. 10? Thank you. What a great timekeeper you are. So we also see an increase in water loss during cooking with ageing. And this is from some data from someone called Shanks et al. 2002. And they did a large number of measurements where they showed on the y-axis is cooking loss, and on the x-axis is the ageing time post-mortem. And you'll see there's a bit of movement around here, but not really significant, so around 20%. But as you get beyond about 10 to 15 days, you get this increasing cooking loss with time of ageing. And this is even though with ageing we know that the water holding capacity of the muscle or the meat actually increases and the fibres actually become more swollen with ageing. So we think we get this increase in water loss during cooking with ageing because you've got a much weakened protein structure in aged meat, and we think it cannot trap or retain the water. So now I'll move on to the structural interrelationships between water holding capacity and tenderness. So the focus when we talk about tenderness is always on the myofibrillar protein integrity and the connective tissue proteins for tenderness. And this ignores, we think, what we call is the third major structural component of meat, and that is water. And that variations in water content of cooked meat are a strong contributor to toughness development. So David Hopkins, who I think is talking next door while I'm speaking, so he won't be in the room. David Hopkins showed that, in fact, when we're measuring Warner-Brasler shear force, we had a big study where we were measuring 2,000 shear forces in lamb per year over a 5-year study. So we had 2 labs doing it, and we had to make sure that we standardised our techniques. And we found there was variation between the 2 labs. In our statistical analysis, we had to adjust for cooking loss. So in subsequent years, we actually used exactly the same water baths, same size, even though we were cooking to the same end temperature. And we also randomised from our sires into each cooking batch because we knew that cooking loss was so important for what Warner-Brasler result you get. So we think this should be adjusted for in statistical analysis and also closely controlled. Now, examples of the relationship between water holding capacity and tenderness include saline injection or marination with both of those. And of course, Eero Palani has done some excellent review a couple of years ago at Icomst talking about the very important aspect of water holding capacity and the role of salts. So certainly saline injection and acid marination cause swelling of the myofibrils, And we know that with these, you actually get reduced cook loss and also more tender meat. And in general, large reductions in water content in dilute polymer systems, such as meat, will increase stiffness and hardness of a polymer system, such as the myofibril. At longest, I'm going to show you something that anyone who's a meat scientist knows really well. At short sarcomeres, you have tough meat. How many long do I have? Sorry? 7, thank you. So we all know that at shorter sarcomeres, we get tougher meat. This was some data that Peter Purslow actually had. So when you have your shorter meat, so shorter than the rest length, you've got tougher meat, and at longer sarcomeres, you have a lower shear force and more tender meat. And certainly the Weaver, who's worked in Dave Gerard's lab, proposed this may be because of access of proteases to myofibrillar proteins. So the effect of the change in the myofilament ladders, although it's quite clear with water holding capacity, why you get more water coming out when the myofilament lattice shrinks, it's not so clear with tenderness, we don't think. So the changes that occur in tenderness with cooking temperature are shown here. This is from Davey and Gilbert in 1974. And there's a number of graphs showing, this is the one we picked to show this. As you go from 30 degrees up to about 100, this is shear force value here. You see this graph that's shown here. So certainly we think in this region here, and Alan Conjoins also proposed this. In this region it's myosin, probably actually denaturing and increasing the shear force. Now this region here is where collagen actually denatures. So it's interesting, you don't see a change in shear force in this region in here. And then actin we think is probably contributing denaturation here and again increasing the toughness. You'll lose the... So with cooking, we get an increasing rigidity of the myofibrillar structure, considerable increase in toughness, and we think this is due to denaturation of proteins and also water loss. So this research is continuing to define specific proteins involved in shrinkage during cooking and differences between muscles. And Roman Buckow, who's in the room, is involved in this research also, going forward. So now I'm moving on to the influence of structure on muscle, colour and light scattering. This is the work of Joanne Hughes in her PhD. So myoglobin content in chemistry is well known, and certainly Suman gave us a great review on a number of different aspects of myoglobin chemistry. But what we want to talk about here is the role of scattering. So much has been written, certainly has been some written about scattering, especially from Howard Swatland and others, but we haven't seen any visualisation and investigation in detail of this aspect. So I'll be showing you some of that now. So basically what we're talking about in terms of scattering is, and the examples I give are here, this is a low scattering medium. So you've got, oops, shouldn't be bending. You've got some particles here, but basically it's low scattering, high degree of absorption, and this would be, for example, high pH or dark meat, which I will show you in a moment. And the example here, it's got more particles, and also the medium's allowing more scattering. And this, for example, would be a low pH mussel, which would be also pale. So the material that we're talking about in terms of where these little dots are could be any components of the muscle, such as muscle fibres or myofibrils or sarcoplasmic proteins. This is now we've used some what's called dark field confocal reflectance. Dark field confocal microscopy. It's not reflectance because we consider scattering. The reflectance word should not be there. So this muscle, this is we have again homogenised muscle fragments. This is under dark field, and it shows all the scattering elements under dark field in a light L star of 35.7, low pH muscle 5.38, compared to this one is from a dark muscle, and you'll see there's very, very little scattering there, and usually these muscle cells are also wider. It's actually got a larger diameter, hasn't shrunk as much. It's from a 24.8 L star and a high pH muscle. So we also see, I'm going to skip over this one, it's actually in the paper, but we certainly think we see changes with high pressure processing, and when we go from 0 to 800 megapascals, and we think the L star value relates most to scattering, whereas, and you see that in these ones here, the L star changes from 37 up to 50, when the A star value only changes from about 17 to 15.5, and then you see more changes down here, and we think the A star value in this case relates more likely to myoglobin denaturation. So we are continuing our research in colour also, and the specific structural elements that are involved in scattering, as well as the role of water. So conclusions? What's that, my time? Good, perfect. So the high muscle water content is important to the structural arrangement of the lattice, and it acts as a plasticizer of proteins. The loss of water from the structure influences the properties of meat, and the majority of research in the past has been on raw meat with a few exceptions. Water loss in raw muscle, I've explained, is myofibrillar lattice spacing, membrane permeability, extracellular space, and drip channels. The cooking process, we see this as different to what happens in raw muscle, involves a process of protein denaturation, shrinkage, aggregation, and water loss. So I think that explains why you don't always get a good correlation between water holding capacity and cooking loss or toughness. The proteins involved in the changes in raw and cooked structures certainly influence water holding capacity, cook loss, and tenderness, and these are all actually pH and temperature sensitive. So during cooking, there is a loss of water from the structure, which decreases juiciness and tenderness. During cooking, the water is influential in generation of toughness. It is influenced by collagen shrinkage. We can't ignore it, of course, but it's also influenced by myofibrillar and cytoskeletal proteins. And the structure of muscle impacts the colour perception and light scattering properties of the meat. So water loss induced by cooking and other treatments changes the structure and influences the surface lightness of meat. So here's a picture of my contributors. Joanne Hughes is working hard doing some high pressure processing work. Here's Sophia Urseth, who made the wonderful movie that we really like. She's doing something with some mincemeat there. This is Peter Perslow, actually came and visited us at CSIRO when I was there for 3 months, and at his farewell party we gave him a football, and Sophia chose a Richmond Football Club scarf for him. So he looks very happy with his what he's been given. And acknowledgements, we certainly had involved in the experiments several people from CSIRO, and we had a very, very busy schedule for 2 months where we had to actually plan every day and who would be available to be cutting up the pieces of muscle. And this will be coming out in a paper in the forthcoming months. So thank you for your attention.